just as an introduction with Wasatch BioLabs, we are a high throughput nanopore sequencing lab. Just like a lot of the other service providers, we help people with all their needs of direct RNA and cDNA and whatever they want. But our specialty and our bread and butter, and really why we're speaking, is our efforts in innovation on methylation-based assay, both in a broad biomarker discovery method and in a very targeted method that assesses a lot of the things we've talked about today. So NESSI-Seq is a term we use internally. It stands for native epigenetic assessment by single cell isolation. So we have come up with a method that can allow us to do methylation analysis in a native way. In a targeted method, we've done it anywhere from, a couple loci up to 10, 20,000 loci, and average about 400 to about 1,000 X enrichment. And we can also do it in a non-targeted approach, both for cell-free DNA and just your general gDNA. We've also done a lot of studies for FFPE-derived DNA, and this is kind of the concept I want to talk about today. So I'll focus less on the methods, more on just some of the results we have by utilizing these methods we've come up with to do kind of robust methylation analysis for biomarker discovery and applying it to some diagnostics. And I'll show you some example use cases for specifically a neurodegenerative disease. So how did we get here? Why did we embark on this effort of coming up with a new method of analysis? So my background is actually in neuroscience. So my PhD is focused on early diagnosis for neurodegenerative diseases. And this is some of the first data we published, which was using a PCR bisulfite amplicon sequencing method on Nanopore, where we targeted 500 base pair regions that were uniquely differentially methylated to cortical neurons. We sequenced those regions and just quantified them in a percentage to show that by identifying cell-free DNA that had a methylation signature unique to cortical neurons, you could correlate it with neurodegenerative disease. And you can see on that box plot there, you've got Alzheimer's on the left, you've got young donors, you've got age control matched donors, and patients with MCI that went on to later be diagnosed with Alzheimer's disease. You've got patients with MCI that never progressed to Alzheimer's and then are purified neuron control. And if you can see the little tiny dots there, the Alzheimer's group and the MCI that went on to get Alzheimer's are significantly elevated to our controls and our young controls. And so this was a pretty exciting paper, performed really well, but has a lot of limitations, not just sample size, but if you want to take a test clinical, you need to be able to have it overcome a lot of the biases that are inherent with the current methods of methylation analysis. So all of these data had really strong PCR bias, bisulfite conversion bias. It was limited to 5 loci. And the epic array tool that we used at the time, the Illumina epic array, limited us to only 900,000 CPGs to be used in our original biomarker discovery. So we were limited in terms of our original platform that we were able to use to do discovery. That's a major problem if you're trying to take it to clinical, right? If you're trying to monitor a patient from test to test, you can't have them come in and get a baseline diagnosis and then try to overcome PCR bias when you're trying to quantify the amount of neuron DNA in the blood. And so we realized pretty quickly that although the research was really exciting, we needed a different platform to be able to actually scale some of these diagnostics clinically. And that's something that's common throughout really all current platforms of methylation, is this really exciting research methods and a limited ability to take something clinical due to the limitations and technical biases. So this is kind of what we came to overcome was, hey, can we come up with a product that allows you to analyze highly fragmented DNA like FFPE DNA or cell-free DNA? Can it do genomic DNA? And can you do it in a broad approach and a very targeted approach? And that's kind of what I want to focus on here. So this is what we refer to currently as native methyl-seq. We're trying to come up with a name for it. But utilizing the ONT platform, we were able to come up with a product that gives you a 30 times more CPG coverage and provides the single molecule resolution that you need in a lot of these cell-free DNA cell of origin analyses. So some of the stats, we average on average, we get about 27.5 million of the CPGs provided at a 5x depth. That's obviously quite a bit more than the 900,000 sites on the Illumina Epic array. It is native, so there's no PCR biases, there's no bisulfite treatment, and so it allows us to give it allows us to get rid of a lot of the technological biases. Something we're really excited about is the flexibility. So you can start off with a broad biomarker discovery and turn it into a targeted approach. And that can be one gene target. It can be thousands of different loci. And we actually do use a pull down method. We do use a hybridization method that I won't get into right now. But obviously it gives the versatility. So we can look at 5MC, 5HMC, and do a positional analysis or regional analysis, and even a single molecule read by read analysis that can be really valuable for things like ctDNA or cfDNA. So in the cell-free approach, the problem we had to overcome is that cell-free DNA is highly fragmented. So when you're trying to utilize the Oxford Nanopore workflows, it needs to come into the pore double-stranded, and if you've got a lot of nicks and fragmented DNA, it causes a lot of problems when you're trying to sequence it through those nanopores. So we came up with a method that fixes all those nicks and fragments that are inherent in cell-free DNA, and then we do take it through a proprietary library prep that's super quick and is optimized for low input. So the average sample that we put through is roughly 5 nanograms or even less, and we're still able to average anywhere from about 100,000 to 1.5 million reads per sample with 96 samples on a flow cell. So I want to dive into later how we can also get really strong genetic information out of it, even though it's considered low input and low pass. A lot of people immediately ask us, how does it compare to Epic array data, even though it's a different data set and a much better data set. People want to understand, how does it compare to data I've already generated with the Illumina array. And so this is a comparison between our native methyl-seq and the Epic array platform. And to make it so you could compare them, we had to limit the regions we're utilizing in this analysis. If you're familiar with the Illumina array, it covers largely CPG islands. That's where it's biased towards. And so this is just a CPG island analysis to show what Nanopore covers for CPG islands, what Illumina covers, and just the confirmation that we are seeing the same data and better data. So if you take all the CPG islands present in the genome, you'll see there that both the Epic array and our platform are able to cover 55% of the same CPG islands. There are 16% of the array that we weren't covering in our 5x coverage, and so we're upping that now to understand, okay, what coverage do we need to cover all of the CPG islands that maybe we were missing before. But then there's 28% of the CPG islands that we covered that the Illumina array did not. And you'll see that there's all versus anything greater that had a differential methylation of above 0.5, because another thing that Illumina does is they really focus on CPGs that are known to have a much stronger bimodal distribution. So if you just look at those, you'll see we both cover about 80% of those. ONT covers 13% more, array covers 7%. On the right, this is just an absolute difference. So understanding the bimodal distribution between fraction methylation on the two platforms. And you'll see on the top there, that's the one I want to focus on, that they're really similar, but you'll see that the array is slightly shifted in on both sides. And that's due to the normalization you have to do on the epic array. And so by default, you lose a lot of resolution as they try to normalize for batch effects. And overall, Nanopore data is just, it gives you a lot more resolution in terms of the individual CPG calls. Where we get really excited is the ability to do a single molecule resolution. So you'll see in this example, we've got two tissue types that are shown on both the array and the ONT platform. And this is really common on the array, where when you're looking at individual regions, you've got 5, 6, if you're lucky, you'll get up to 10 or 15 CPGs across a region. And in this case, you can see there are hundreds of individual CPGs in that island. And if you look on the top line of the array, you can see they're highly methylated in one tissue and low methylated in another. On the array, you would think that was a perfect region and that the entire region is methylated or unmethylated. But when you look on a single molecule resolution, you can see there's a lot more gray area. There's a lot more sites that are methylated in one and unmethylated in the other. And so having that single molecule resolution is really important, especially when you're trying to characterize tumors or any other cell-free analysis on a read-by-read basis. One of the exciting things about this is we can look at it in both a 5-HMC method and a 5MC method, which you can't do on the array. So coming back to neuro, so we took this application and now came back to our neuro project to see how we could improve it. And we started into a 500 sample study. And the whole goal of the study was to differentiate between Alzheimer's, Parkinson's, and ALS. So A, show that we could identify a signal. We could differentiate disease versus non-disease. And then B is to differentiate between the disease states. And to do that, we first had to rerun our original pipeline on the new platform rather than the epic array. And you can see the comparisons there. So as we did our models to identify our biomarkers for cortical neurons on the epic array, we started off with roughly 9,000 predictive sites that were able to differentiate between cortical neurons and anything else in the bloodstream. When we run it through our new method, we average 2.4 million predictors. For dopaminergic neurons, we went from 5,000 up to 843,000, and spinal motor neurons 2,000 to 413,000. So hopefully that ingrains in your mind that there's a lot of biological information that we are just flat out missing on current methylation platforms. And by utilizing ONT, you can get a lot of insight into signal that otherwise you're just missing. One of the exciting things is you can do this a lot at a significantly cheaper cost than the epic array. And this current project, we're optimizing to create a kind of a one-size-fits-all blood test to diagnose all of these diseases. Now for cell of origin quantification, the next question we get is, all right, how low can you go with the Nanopore flow cells? And so this is a downsampling report where, I just noticed in there, it says 100,000 million reads. That would be really impressive. Not 100,000 million reads, 100,000 reads per sample. And we downsampled all the way to just 1,000 reads per sample. And you can see on the right there, we have the predicted fraction versus the actual true fraction of that spike, and we did. And we have a really tight R-squared, so we can get down in the hundreds of reads and still have a very strong predictor of the cell of origin. When you get into the genetic side, we still see a very strong ability to utilize this information for your traditional copy number variation, structural variation, and single nucleotide variants. This example is a situation where each sample had roughly 0.3 to 0.4x coming off of our platform for methylation. We had 4 males and 4 females on this particular run, and just went and use them to show that we could spike in various levels of DNA and accurately predict copy number variation. So you can see on the left there in the blue, we've got 100% XY for the male, 0% XX, and we're able to clearly, you can see it step up in each dilution where we added various ratios of DNA all the way up to 100% XX, and we can clearly differentiate between those chromosomes. There's a lot of work to be done with copy number variation. Our expertise is in methylation, but we have done some work to show that you can utilize both the genetic and epigenetic information together. In terms of making it useful for a lot of people, our clients, we want to enable them to do biomarker discovery rather than handling it all ourselves. And so our team of bioinformaticians have put in a lot of effort to make this an open source product that anyone can utilize. So either you don't know what you're doing for bioinformatics and you can just follow the README and input everything line by line, or you can edit it to do any of your own unique analyses. The way we've designed it is you can do anything from methylation summaries by position. You can do CPG islands or really any region you want by just uploading a bed file into the code. You can do regional analysis. You can do a read by read analysis. You can even make it match the Illumina array. You can do sliding window analysis. Basically, we've just enabled all the tools that allow you to do very basic methylation workflows to get the same power out of this data that we can. We've also optimized our outputs to be in very clean databases that allow you to compare your current data and your data to previous workflows or assays that you've used, like the Illumina read. Here are two examples of what those databases look like. So you have a positional summary on the left, and you can see in this specific case it combined both the MC and the 5-HMC into one column, the MH counts column, and it just gives a fraction methylation score of each position in the genome. And then on the right there, it is a summaries by region. And so this person's picked specific regions to do their summary. One of the things that is in the background that a lot of people don't realize is we've designed this in a way that it can be run on any laptop. A lot of times when you're doing methylome-based stuff, it tries to burn a hole through your desk. And so all of these runs, these two runs specifically, were pushing 50x coverage, and they did an entire positional summary of all 28 million CPGs in a sample set of 100 in like 48 seconds. They were able to get the entire thing completed just on your regular M2 MacBook. So in terms of applications and future looking, again, we put together this platform to enable people to innovate on it. And so I showed the example of neurodegeneration and what is being performed there from a cell of origin method. There's a lot of work being done that we're helping for oncology, infectious diseases, fertility, forensics, and many more. Some of those are risk scores, some of those are biomarker discovery for companion diagnostics, and some of those are just breakthrough diagnostics that they're trying to take to market. With that, I want to acknowledge all the people that put effort into this. I am actually a PhD student at BYU as well. We did a lot of this work through Brigham Young University. Tim Jenkins and Jonathan Hill are my co-founders. KU Alzheimer's Disease Research Center has been a big support as we helped scale the neurodegenerative project, providing samples, key opinion leaders. A lot of the funding is coming from Renew Biotechnologies, which is a parent company to Wasatch BioLabs, owns a majority of the shares. And then obviously Oxford Nanopore couldn't pull any of this off without them. So thanks for your time.